Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled From Process Development to Manufacturing: How to Successfully Optimize and Scale Up the AAV Enrichment Step on a Novel CIMmultus QA HR line. I'm Charlotte Barker, an editor at BIA Insights, and joining me today are Rok Gigon and Maya Leskovitz, who will explore novel monolithic columns that allow for batch-to-batch and scale-to-scale reproducibility within 3% of the AAV8 empty capsid iso-conductivity elution. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to Rok and Maya using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics we're covering today. So now I'd just like to briefly introduce our presenters. Rok is Head of Product Application Area for AAV and Adeno in the Process Development Department for Viral Vectors and Vaccines at Sartorius BIA Separations. He has 6 years of experience in the field of downstream bioprocess development for gene therapies. Maya is heading process development for viruses at Sartorius BIA Separations. She has more than 10 years of experience in the field of downstream development, working in the laboratory as well as leading development projects for clients. So without further ado, I'm going to hand over to Maya to kick us off. Over to you, Maya.
Thank you very much and welcome everyone. It's a pleasure to connect with you today. As we are excited to introduce our innovative CIMmultus QA high reproducibility line, a game changer for AAV manufacturing and process development. Our webinar today will be split into 2 parts. I'll first provide a snapshot of our company and the products, and then Rok will present a case study on optimizing AAV polishing step with the novel QA HR line. Thank you very much all for joining us. At BIA Separations, we are an exclusive producer of CIM monolith columns. With over 25 years experience in purification of large biomolecules like viruses and nucleic acids, and also with the Sartorius acquisition in 2020, we became Sartorius Center of Excellence in Gene Therapy, offering solutions for downstream process development, manufacturing, as well as analytical methods development. The comprehensive product portfolio covers preparative chromatography columns designed for efficient purification processes, high-throughput screening devices, and analytical solutions, including our CIMac analytical column and the PatFix platform, tailored for precise monitoring and control. All of these offerings is underpinned by the monolith technology, very well known for its high flow rates, high binding capacities, and high yields. Furthermore, our offerings are complemented by cornerstone biomanufacturing development services, which we will explore a little bit more in detail in the upcoming slides. We offer short feasibility studies that leverage existing technologies and expertise. These studies are conducted without extensive optimization or robustness assessment, making them ideal choice for clients that would like to learn about the technology capabilities in a short time. For more comprehensive needs, we offer process development that include complete chromatography procedures, from analytical methods to the process itself. We deliver detailed standard operating procedures and facilitate seamless technology transfers. Downstream development for AAV is one of our core expertise. We have downstream process solutions that cover all serotypes and utilize tools to achieve high yields and exceptional purity. Optionally, we can extend our services to include upstream part as well. The team of upstream experts equipped with advanced analytics aims to maximize both titer and percentage of full capsids right from the harvest stage. The schematic outline of the AAV purification below in the slide highlights the stages where we really concentrate our efforts. So specifically on the capture step and the chromatographic polishing step. Our approach is data-driven, employing design of experiments for condition optimization, followed by process verification, where we define critical process parameters. We also conduct robustness check to ensure the reliability of the process. Once these steps are validated, we proceed with the scale-up and technology transfer. Experiments at Sartorius BIA Separations are conducted on columns up to 80 milliliters to ensure that the process is fine-tuned before and scaled up before it is handed over to client or to a selected CDMO for full-scale production. The true measure of success of any process really relies on the analytical methods employed. Our process development integrates both developers and analytical experts within the same team, basically often working side by side in the same laboratory, on the same bench, to expedite the achievements of our objectives. The core of the analytical strategy is focused on leveraging PathFix analytics, complemented by a range of orthogonal techniques, including PCR, mass photometry, ELISA, and others. The orthogonal approach really ensures process understanding and provides confidence in the developed procedures. Worth to mention at this point and say thank you to many of our esteemed partners that already collaborated with us to refine their processes, ultimately guiding their products to successful market launches. And if we take a minute to reflect back on the evolution of our field, on the AAV field, I think it's remarkable to consider that the use of monoliths for the separation of full and empty AAV particles was first documented in the year of 2012 by Martin Locke from UPenn. In this decade since, over 20 articles have been published on the subject, illustrating the growing interest and the advancement in the area. AAV vector is now really a cornerstone in gene therapy, with several thousand clinical trials currently underway and 8 drugs having received the approval. At Sartorius BIA Separations, we've contributed to the progress with over 60 AAV purification projects and several publications. However, I would just like to draw your attention to 3 key developments from our company in the past 6 months that stand out, which is improved methods with the high resolution of AAV empty and full, high throughput screening technologies the second, and the third one was the launch of the CIMmultus QA high reproducibility line that really represents a significant leap forward in our capabilities. These milestones really underscore the commitment to innovation and our role in advancing the field of AAV purification. Now, just a few words about the QA HR line. The innovation here really lies in the shift from the traditional column testing methods to the ones more suited for the unique challenges of AAV empty and full separation. Originally, columns were tested and released based on their performance with small molecules, such as using phosphate to measure the ion exchange capacity. However, during the years, we found out that there was little or no correlation between the ion capacity and the conductivity at which capsids elute. And recognizing this, we sought a more appropriate analytical method to ensure that the columns would be compatible with the AAV processes and maintain consistency across batches and scales. This led to the development of a release testing protocol that is specifically tailored for AAV separation. Initially, our task was to create a representative specimen. First, we start with the polymerization and modification of the parent monolith, as seen on the left side of the slide. Then follows cutting a 0.2 milliliter disc from both the top and the bottom rings of the parent monolith. The monolith is finally encasted within the housing of the CIMmultus line. This is now our packed column. And each specimen is packed separately in a small housing. Our research and development department conducted multiple tests to confirm and to demonstrate that the specimen really accurately represents the performance of the packed column. Each specimen is then separately tested to guarantee the high reproducibility of the packed column. And due to the fact that we didn't find appropriate and representative molecule, each column within HR line is now released based on specimen measurement of AAV8 empty capsid elution conductivity. On the left-hand side, we see a comparative analysis of tested columns ranging from 1 milliliter scale all the way up to the 8-liter column. And it's really reproducible, and this really demonstrates our commitment to quality and to consistency across the entire spectrum of product line. Release criteria are stringent, with a tolerance of only plus minus 3%. This stands a new benchmark for batch-to-batch and scale-to-scale reproducibility in chromatography. In other words, processes developed at small scale can be seamlessly translated to manufacturing scale when using the QA-HR line. Worth to mention that success of entire process is also dependent on reproducibility of other variables, such as buffer preparation, temperature, sample, and so on. With that said, and without further ado, I'll pass the word to my colleague Ruk, who will discuss this further in the case study presentation.
Thank you, Maya, for the introduction, and greetings, everyone. So, starting my presentation, I would like to start with a statement, or rather a fact. And this is that the scaling up should be easy. And I think you all agree with me, but to look at really from a reality perspective, well, scaling up is not that easy. So we have to account for sample variability, temperature that is different on the process development scale and also in the GMP suites, and the pH and conductivity shifts in buffers, the loading ranges, maintaining appropriate flow rate, in the end also matching the size of chromatography skid to the column, and lastly, as it is bolded here, so the chromatography media variability. And our, let's say, answer, the Sartorius BIA separation answer to that, at least for the last statement, so the chromatography media variability, is really the novel CIMQA HR, which stands for high reproducibility line, that I will go through in this presentation. So the agenda for today is really we will go through anything how you design from the process development state and then up to the scaling up into the manufacturing. So we will start with the selection of polishing column, and we will use the showcase of our three toolbox or three columns that we use, which is the QA HR line, the Prima S and the Prima T column. We will show then on the second point of the agenda that we can do the screening using either the high throughput format or by a single column application. Then the capacity runs, so how we determine the linearity and why the confirmation is needed. After that, we will continue with the formation of step washes, so how we can design the actual run, so by either bind dilute mode or the flow through mode. And finally, also wrap everything together with the scale up from our smallest unit, which is 1 ml, up to the 8,000 milliliter column, so 8-liter column. So if we start with the first one, which is the selection of polishing column, I will also bring up the pointer since it's easier to navigate this way. Basically, what we wanted to first to do is use the same sample for the entire study. And this is here, the first slide represents the initial sample characterization. So what we started with, which was our core sample. And it is HEC 293 suspension-based AAV8 serotype, which was pre-purified with TFF, which was coupled with DNase and captured by our CIMmultus SO3 column. Because as said, we wanted to have the entire same batch for testing the entire from screening up to the scale up. We prepared 7 individual upstreams, which were then also followed by the mentioned downstream. And in the end, we totaled with the 7 E15 vector particles of, let's call it purified but not enriched AAV. And this sample was then analyzed here as seen by 4 orthogonal analytics. If we go to the left side to the right side, so the first one, we have the PET fix coupled with our analytical CIMac AAV column. And what we can see here is we have the 3 detectors. The top one is the UV absorbance, which is the 280 by blue and 260 by red curve. In the middle, we have the tryptophan fluorescence, and in the bottom, we have the multi-angle light scattering detector. Because CIMac AAV is a strong anion exchanger, it separates based on charge. So the empty particles will elute first, the fulls will follow, and then also we use the fluorescence for the estimation of the ratio, and this is here color-coded or labeled in light blue color. The next one was the PET fix UC. UC stands for ultracentrifuge. This is the approach that also we do in-house, and it is a preparative ultracentrifuge with cesium chloride, which is then coupled with our PET fix PET fix apparatus that basically then has all of the detectors, and it picks up the different signals. What I'm showing here is just the fluorescence signal, so the middle one here, and in opposite to the CIMac AAV, here, because it's a density gradient, we go from a more dense environment to a less dense environment. So the fulls will elute first, and then they will be followed by either partials and then also the empty capsids. The third to the right is then the mass photometry, which is using the CIMmux MP machine, and it separates based on mass. And again, we see the empties, barely any partials, and then we have also the full capsids. And on the far right side, we have the charge density mass spectrometer, which is done by the Megadalton solution, and we also have it's based on mass and charge. So we have the empty eluting first, then the red portion is the partials, the green ones are the fulls, and then the oversized population would be the blue ones. Those are all of the graphical representations, but if we put them in a table, and also if we account for the dPCR and ELISA, we can also come, let's say, to the most commonly used method for empty-full estimation, and this is the ratio dPCR versus ELISA, so genomic titer versus physical titer, and we are averaging around 25.3% of full vector. And if we look across other analytics, we do see that they are more or less really in line. So everything around, so the lowest number was 21% up to 25%. So really, although they're all orthogonal, so having different approaches for the estimation, they really do show the same picture. And this is something that you want to see in your analytics. So if one would be outlier, that means that something is wrong with your analytics. But if all are pointing into the same direction, then this is a good prediction. So first in line was really assessment of what is the most suitable column. And as stated, we used 3 of them. So the QA HR, the Prima S, and the Prima T. How do they differ? So the QA is a strong anion exchanger. Prima S and Prima T are both weak anion exchangers. In that difference, that the Prima T also has metal chelating effects, so we can exploit those traits. But how, let's say, the means of elution, how we get the elution from the column. For the QA, we use the ascending salt gradient. For the Prima S, we use the pH gradient. And for Prima T, we also use the ascending salt gradient. But the salt can vary. Why I have 4 figures and not 3 is that also, although we're discussing the QA HR, we are offering also now a novel method, which is now being patent pending. So we cannot disclose the conditions for it. It is part of the cornerstone services that Maya mentioned. But the conventional method here is the regular one that is also can be used. The conditions could be found on our website. So on all 3 columns or 4, let's say, approaches, we loaded the same amount, which is, depending on the vector genome or vector particles, around 2e13 vector particles per ml of column. We used 1 ml columns for each, and you can see that, let's say, the profile really, so it behaves differently. So we have the empty capsids eluting first, then the full capsids, and then the remaining portion of, usually we associate them with aggregated or damaged capsids. On the novel method, we do see that we really increase the resolution between the two subspecies, and Prima S and Prima T also shows a different profile. So the main conclusions are then down with the analytics, and this is showed on this second slide. So we assessed all of the main eluates. So the main eluates were the main peak that eluted from the 3 mentioned columns and the 2 approaches. 4, we assessed them for recovery and for percentage full. And for those 2, for recovery, we used the dPCR estimation and also PETFIX CIMac AAV, which is also capable by integration of the peaks. And for percentage of full, here the same method simultaneously gives you both a reading of recovery, but also you can determine the percentage of full. And the another one for percentage of full was the mass photometry. So if we look at the data, we see that the highest enrichment and recovery of full AAV capsids was observed with novel QA HR line. And also the highest was also achieved with the novel method, which is the yellow bar. And another conclusion that also could be made here, so we're seeing that if we compare the light gray, which is the standard, the conventional method, versus the novel approach on the QA, we do see that the recovery is maintained, but the purity or the percentage of full really increased depending on the analytics, but up to those 20% increase. For the other 2, so for Prima S and Prima T, they're, let's say, we could say that they are maybe suffering on the recovery, especially the Prima S, but in regards to the purity, they really deliver also the same quality of the product as the QA. But because we are doing conclusions and averaging, we want to have 2 variables that are fitted. Then we advanced with the QA HR line using the novel method. So now that we selected the media, we want to do the screening design. And this is, so here I will present the 2 approaches that we recommend and also we offer in our portfolio. So the first one that is shown here is by high throughput screening using the 96 well plates coupled with our PETFIX system and also the analytical column. So we loaded 1E11 per well of the plate, and we use the microplate reader in addition also to the PETFIX system. So here is the experimental scheme for the conditions that could be tested and what we used. And you will see that I'm stating here the eluting agent A and eluting agent B. Since we're not disclosing due to the patent being filled, I cannot show those. But basically what was then also assessed was different pH ranges going from neutral ones to the pH 9. And then here on the top part, we are increasing in salt concentration. So we are figuring out where the vector binds and then which concentration of this eluting agent is needed that it displaces the AAV. The data for this is shown here on the next slide. And I had it divided on 2 sections. So the first one here on the left is how it is seen by a regular microplate reader, which is tracking the fluorescence signal. And on the right side, I have how it is the same results, how they are seen with the PETFIX system coupled with CIMac AAV column. So it is also divided then horizontally on the eluting agent A and eluting agent B. And if we focus first on the microplate reader, what we do see is basically just the color coded is so the different colors represents the pHs. And then on below, we also see the eluent concentrations. And what we can conclude is that if we focus on the eluting agent A, that roughly around 15 millimolar concentration we do start to see some elution of AAVs, but we don't know really if those are empty eluting first or full eluting or both together. And we just see, let's say, a cumulative profile elution. And we also see that, let's say, for the pH 7.5, roughly around 40 millimolar, the majority of vector elutes. So further increasing in salt would not really change much. But also what can be seen that the higher the pH is at, the higher the salt is needed. And this is also confirmed by theoretical approach. So we are with higher pH, we are farther away from the isoelectric point. So we need, the AAV is in that way more negatively charged, more bound to the QA. So we need higher salt to displace it from the column. The same is for Elu and B, just the different concentrations were used, but really no concrete conclusion could be done is where my empty solute and where my full solute by using the regular microplate reader. This is where PetFix really comes in handy, and basically seeing just, I will be showing due to the, let's say, amount of information, just the pH 8.5, so which is here the blue color. And we are having a fluorescence detector, how it sees the signal from in those different concentrations. And what we can see for the eluting agent A, that from 20 millimolar, nothing yet elutes. Then at 25, we see empty capsid start eluting. At 30, also empty capsid still. And then at 35, we already see that some of the fulls are eluting. The same thing could be also said for the eluting agent B. Just in this case, we have let's say a more narrow window of opportunity. So we are seeing that between 20 and 30 millimolar, we have either just empty eluting and then also entire full eluting. And also, if you compare the two eluting agents, we do see that in case of the eluting agent B, we already eluted the entire fulls, where here they are still starting to elute. And that means that the eluting agent A has a better selectivity, or it's more appropriate for our process development in that regard, that there is a better resolution because it elutes, it has more window for setting of the step wash, meaning also that then we can then easier implement this in our step wash approach. Same thing could be also done with a different approach, so that we load it per column and we do a single column estimation. And in this case, we went with the 1 ml column loaded at 1E13 vector genomes per milliliter, and then we increased our eluting agent A, in this case, by gradual increases. And we can see that it starts eluting around 46% of pump B, and the full starts to elute around 70%, and then also it lingers on a bit. If we translate those two numbers in millimolars, so what is the concentration of salt, we do see here in this top table that 46% B equals 25 millimolar eluting salt, and 70 is roughly 38.5 millimolar of eluent A. And when we compare those numbers to what was shown on the previous slide with the 96 well approach, we do see that the data is in line. Basically, we get the same conclusions no matter if we go with the 1 ml column or 96 well plate. We do see that the same starting point of empties will elute. For the fulls here, it is not exactly the same, but this is also, we could go with more steps in between and not 6% increments as were done here. So we would be closer to the number here. So the takeaway message is really that no matter the options that you choose, the results will be in line, and you can really well predict your pH, so binding conditions and eluting conditions. So now let's say we are still in the process development. We selected the column, we selected what is our conditions for binding, also for dilution, but now we're interested how this affects when we are dealing with different challenging of the column, so how much we load per column. And in this case, because the high capacities are reported with the use of the CIMmultus columns, there is a really great tool that also Maya went over, and this is the specimen. which is the monolith extract from the parental column, and it exists in 0.2 milliliter bed volume. So it's a perfect fit for such applications where high sample consumption is seen, and we can really reduce the amount of sample per getting the results. So what we've done is we loaded in different amounts. So we loaded 5e12, or maybe I will be talking more into the vector particles because the capacity is dependent on the particles rather than the genome. So we loaded anywhere from 2e13 up to 2e15 vector particles per milliliter of column. And in the last case, so the red curve, we also, it is not shown here, but in the flow-through response, we reached a breakthrough, meaning we had some of the empties present in the flow-through, and that also manifests on the chromatogram. We could see that gradual increase in loading will shift the elution profile, will migrate to earlier retention time. But if we surpass the capacity, as we've done with the red curve, so at 2e15 vector particles, empties were already present in the flow-throughs, fulls were still bound, but we do see that we completely lost the resolution, and also we are far away from, let's say, the starting point of eluting salt. So what is the apex of the peak? Here is also this then represented in the table, so how the conductivity looks at different stages. And usually during the process development, we do such a estimation, so to determine what is the, let's say, the lowest amount, the LOD, and also the capacity. But then we ask our clients what is the, let's say, the scale that they will be at, and usually we see that their loadings are around 1e14 vector particles per milliliter range. So we took this one as a benchmark, so as a reference point. And then what we have to see to get where we are in linear response, and the HR line offers 3%, so that all the values fall within 3% margin. So then we see that the two values, so that one here and also the next one, are falling in this region. So really the linear loading range within those 3% margins should be from 2e13 vector particles to roughly 5e14 vector particles per milliliter of column. And this was done on a specimen, but we always encourage you to also do the actual run on the larger scale, or maybe also do a confirmation run on a 1 ml scale. And we've done just that. So we loaded the lowest amount, which is 5e12 vector genome, or 2e13 vector particles, and the highest, where it's still linear, so the 5e14 vector genome. We went with the linear approach, so linear gradient approach, and we collected E2 fraction, which is the empties, then the E3 fraction, which is in between the two, and E4 fraction, which corresponds to full. And we assess those by, again, the same analytic as before, so PCR and percentage of full by mass photometry and PET fix. The graph represents, so the bars are showing the two, so the lowest, so the low point of loading and the highest point of loading, and the step recovery, it's aiming just at the main eluate, which in this case is E4. So in E4, in both approaches, we're reaching roughly 70% of vector recovery. The mass balance, then the next section, shows us what is the cumulative, so basically the entire recovery, if we account also the flow through plus wash, the E2 and E3, so everything that comes off the column. And in this case, we're seeing full recovery. So from those 93% recovery mass balance, 70 are of interest, so of our main product. How it looks for the percentage of full, we see higher numbers showing with PET fix, and slightly lower with the mass photometry. But the main takeaway here is that no matter the scale, no matter the challenging of the column that you go, so low amount of loading or high amount of loading, we do see that basically the same values will be obtained. Looking closely more also at the figures of the analytics, not just expressed in numbers, we also get a hint of what the E3 fraction consists of. And it's usually, this is a decision from when we're doing the process development. So usually the valley in between contains some vector genome, and it's our decision, do we want to pull this together with our main fraction, or do we want to put it away? And usually those kind of figures then come in, they basically provide an answer for us. And here we can see that the E3, it's neither empty nor full, but it sits somewhere in between, based on the PETFIX analytics, which sees the charge differences. So it is, we might call it, let's say, a subpopulation of AAV with a higher charge characteristics than the empties, but lower than the fulls. Whereas for the mass photometry, we also see the E3 is in between, and we can see, if we would take a vertical line, that the mass of this fraction is slightly higher than the mass of the empties, but lower than the mass of the fulls. So usually we treat this E3, although it might have, I don't know, 5-10% of vector genome in there, we usually treat it as a product-related impurity. It is AAV, but because we want the purest product that we can achieve, we rather sacrifice it and leave it behind, so not being part of the main pool. The last table on this slide also shows that the comparability between the specimen and the CIMmultus column, and this is again those confirmations. So is the specimen a reliable tool for my column elution behavior determination? We do see that if we compare the conductivities at, let's say, the full apex of the peak, we do see that both specimen and CIMmultus, they're in accordance. So the difference between the two is less than 1%. So whatever was seen, let's say, on the previous slide, we can tell for sure that also we will see profile-wise, we will see also on a larger scale. So now we covered also the challenges of the column, and now we go into the approaches for the step washes. I'm presenting here two approaches. One is the bind dilute mode, which is the most common, and another one is also flow-through mode. So the bind dilute is self-explanatory. So we bind the AAV, and then we use a couple of washes to displace first the empties and then also the fulls. The flow-through, on the other hand, is we went with the approach that we matched the conductivity to that stage that the empties are present in the flow-through, and then we have just the elution with this, let's say, E3 fraction that was seen before, and also then the main fraction. Again, no matter if you really do your buffer preparation as it should be, we are seeing that the step recovery, the full percentage, and also the full percentage on PETFIX, they are in line either with buffer exchange approach or with the flow-through mode. So we're seeing same numbers no matter the approach that you go. One thing that I would like to emphasize here, and I see a lot of struggles, especially when scaling up and switching, let's say, from PD to GMP suites, it's that prepare your step washes not on percentages, but on actual concentrations, and this will be also presented on the next slide. So we estimated what is the 40% of our buffer B and buffer A combinations, so what the actual concentration of elutant is there, and we prepared buffer with exactly that concentration. Also match the temperature when preparing the buffers on the working range that we will be doing your manufacturing runs, because majority of those runs where we're dealing with anion exchangers are using the temperature sensitive buffering system like TRIS or BTP. So definitely take this in account, that you have to temperate your buffer prep to the temperature that you will be running your process. And this also is then has to be also taken into account the constant titrant mass. So do not prepare your buffers on volumetric scale, but really on a mass scale, so that you always have the same amount of the titrant in there. Finally, we come to the scale up. So really to wrap everything together. So from process development, now we're going into the how we implement the step washes and doing the large scale up. So we used the 1 ml column, 8 ml column, and 80 ml column as they are, but because based on the capacity that I've shown, we would need really a large amount of material for the 800 and 8 liter scale, and we've shown that the specimens are reproducible and show the exact values as the parental columns, we use the specimens for the two largest columns. We loaded all of the columns 1E13 vector genome or 4E13 vector particles per ml. We used two different systems, so the Ectopure 25 and Ectopure 150. And as said, we went with the flow-through approach where the washes were predefined, meaning that we had the known concentration of eluent and not based on percentages. So we loaded in such concentration that it displays the AAV in the flow-through, which was 27.5 millimolar of eluent A. That also brings up the presentation that I showed that roughly around 25 millimolar, the empty starts to elute. And then we done a couple of washes. So the A is using the same as buffer A. The B has slightly higher concentration, and the C then elutes our main peak. And what we can see, so here's the full chromatogram, and then we also have the zoom in section. We do see that we collected, once the step started, we started the collection for 4 CVs, and entire AAV eluted in those 4 CVs. We can even go with 3.5 or even 3 CVs to get a higher concentration factor here. And we do see some, let's say, shifting left and right, but also if you look at the conductivities, those are really then in regards to the system being used and not the actual columns. But the main thing here is really, if we take the 4 CVs, everything really eluted within this window. If we look at the data, we assessed it by vector genome recovery and percentage of full. First, we'll focus on the vector genome recovery. So we've done 3 replicates per analytics and also per column. So we have it then selected show by 1 ml column, 8 ml column, 80, and then 800 and 8000 ml column. The error bars are showing the deviation in the replicates, and then the light gray color is PCR. The middle one is the PETFIX, and also you can estimate the titer from the mass photometry. So we're showing all of those, how it shows on the different detectors and on the different columns. The table below then averages entire scale. So if we take in account, if we average anything from 1 ml column up to the 8 liter column, we do see that we are having roughly 75% by PCR and anywhere between 79-78% by the 2 analytics in the entire scale. And also what we can see are the CVs. So the highest CVs are seen with the dPCR, which also you can see here, the large bracket. But basically the consistent elution profile also matches up with the readings for the recovery. On the other hand, we also having then the percentage of full capsids. In this case, we added also an additional analytics, and this is the charge detection mass spec, which is the last in line, the gray color. And also here, we do see that the PETFIX is showing somewhat a larger percentage because it cannot fully separate the empty and the full, so there is still some overlapping of the two. But the other 3 analytics are showing basically the same values, no matter the scale that we look at. So we are having anywhere from 99 or around 86, 87 for the PETFIX ultracentrifuge and CDMS, and slightly lower numbers for the mass photometer with their representing CVs. The conclusion here is also then conjugated, so it's taking everything together. So here we have the average across scale, but then also we've done average across analytics. This is something I would not encourage you to do, but just for the sake of, let's say, having a single conclusion and let's say a take-home message, what is my recovery and what is my percentage full looking. So we're roughly 77% we reach 77% of recovery with a purity of 88%, accounting all of the analytics. Also, we've shown that the scaling up from specimen to parental column is comparable, and this is shown here. So the solid line represents the parental column and the dashed line represents the specimen. So we've shown in the instance of the capacity estimations, but also here we do see that the actual profile when we are doing the step washes, also on the data, we're seeing the same number, no matter, also if you go with the specimen approach. So really, the specimen is a representative for a CIMmultus scale. And lastly, I started with the orthogonal analytics for the feed sample, but now also for the polished sample in the case of ATML column. So we started with roughly 25% of purity for the full vector on the capture step, and now we're seeing the same analytics, but on a polished version, and we are having anywhere from 100 up to the 86 or 75% purity, depending on the analytics. So the conclusions here are that from the 3 polishing columns, the best recovery and purity was observed with the HR line using the novel method. We showed that basically you can either go with high throughput options with 96 well plates, or also with the preparative columns to get to the same evaluation of buffer exchange, of bind-elute conditions. The specimen is a reliable and great tool for both capacity determination and also elution profile behavior. CIMmultus line supports broad range, loading range, so from 2E3 into 5E14 vector particles while maintaining the same recoveries and enrichment. Both bind-elute and flow-through approaches are feasible to be used with the novel method. And finally, what, let's say, the presentation is all about, the CIMmultus now really enables the use of the step washes across entire CIMmultus preparative scale, so from 1 ml up to 8-liter scale, achieving the same results. And with that, I would like to thank the entire team. So not just the process development where me and Maya are coming from, but also the upstream process development, the research and development for monoliths and manufacturing, which really put a lot of effort into launching the column of, HR column, and also our product manager and managing directors. So with that, I'd like to invite you to our monolith symposium, which will be held in a couple of weeks now in Porto Roche in June. And with that, I would like to take your questions.
Brilliant. Thank you very much, Rock and Maya. Great presentation. So as you said, Rock, we're now going to start the Q&A. So if you could please now stop sharing your screen and join me on camera. We will take some questions. Do keep your questions coming in, everyone. We will get to as many as we can and answer any that we don't get to today by email. Okay, so the first question I've got here, what monolith pore size do you use for process development on an HPLC and FPLC? And they've said ACTA Avant.
So yeah, I can take this one. Most of the work that we do in-house is that we use the 1 milliliter columns. In some cases, also the smaller one, because we like to spare with the sample consumption, and this is usually like really valuable. But then anything up to like, let's say 4 milliliter, 8 milliliter, up to 80 milliliter, I believe it's possible to run it on a normal ACTA system.
Brilliant, thank you very much. Is there a difference in purity and recovery when comparing performance of traditional QA with the new QA HR columns?
Right, I can take that one. Do I have my, I'm not seeing right.
That's okay, Rock, just keep answering. We can always look at Maya even if we hear you.
So the difference of the novel QA is really what you benefit from using the novel QA. So the HR line is this implementation of step gradient. So meaning you will have a consistent elution for the AAV specifically, which really has there is a small discrepancy and small difference between the empty and the full. Really need high reproducibility of those 3% to match those across the scale. But in regards to, let's say, process development, if I will maybe answer in that way, that if you already started with a regular QA and you have the data for the purity and recovery, those will not change. So the profile will be the same, the recovery will be the same. The only thing that you gain with the HR line is really having those scaling up options and really now implementing also the step washes rather than relying on a linear gradient.
Perfect, thank you. When you perform linear salt gradients, do you bypass the inline mixer?
So for the linear salt gradient, we never bypass the mixer. So it is usually used for the captures. So for the step washes, what we do on ACTA, so we use the step and fill. So meaning it does a pump wash, and then you put it across the column. So usually I recommend that you put, maybe you can switch with a lower mixer. So instead of, 5 milliliter, you go with 1.4 milliliter of volume, but definitely keep it in there. Just match the fill, so the fill in method rather than having a T-cross or something in between.
Brilliant. Do you orient the monoliths with the flow direction pointing down or up?
Yeah, that's a really good question, because it's actually very important to have the correct flow path, so the correct orientation of the flow direction. The bigger units are usually operated from the lower part upwards, but what is really important to keep the flow path in the direction that is marked on the column. So on each column is an arrow, because it's a radial format, and flow of the liquid goes first on the outer side of the monolith and then crosses through the monolith, and on the inner side it's then the outlet.
Okay, so don't ignore the big this way up arrow.
Yes, exactly.
Good advice. Okay, so it's quite a long question here, so I might break this up into a couple of questions. In FT mode, when you load samples at high conductivity, are you sure that the loading process can achieve the same resolution as the bind and loop modes, sending only the same percentage of empty to the FT?
Mm-hmm. Maybe I start with that one. So definitely, so this is something here I presented, let's say the example on AAV8 using a GFP also as a let's call it GOI. But if you change, let's say, either the capsid or the gene of interest, those conditions will change, and this is why you have to do the development. But basically, you have to find a point where your empties are eluting, but not your fulls. Your fulls are not eluting yet. And if you see that point and you match it, then it will be a constant elution profile across the scale. So I would have quite a strong faith that it will work. The bind elute is just, let's say, comes more feasible when you really have, let's say, you're working on a lower spectrum, and this is, let's say, notorious for AV9, where you really need to have low percentages of salt in there. We're talking 2-5 millisiemens. So sometimes ACTA can do, when it's triggering and doing pulse switches, there can be a shift of conductivity. And in these cases, maybe it would be a better fit to go with a linear gradient, because those spikes could eventually also displace your AV. But if you're above that point, so I would say anything above 5 millisiemens, then it's just a choice of preference.
Perfect. Is QA HR already available in all sizes, and is also available with CGMP columns, and what's the delivery time on that?
Yes. So yeah, thank you for this question. It's available in all sizes since last, I believe, October 2023, as well of course, in a CGMP format. So basically from 1 milliliter column up to 8 liter column. And the delivery time for the small units are basically on stock. But then for the bigger units, it's a few weeks, I believe, the lead time. But you can always reach out to us and check the current delivery times.
Great, thank you. Do HR lines also exist for other modalities, Prima S or Prima T, for example?
Mm-hmm. Yeah. So for Prima S, it's already available. For Primatic, however, it's still a beta product, but it will be launched we're talking now in June or July. So basically, we're also aiming to get as soon as possible to the market the HR line. So if needed, just contact us, and also we can arrange something prior to that deadline.
Great. I think we've got time for one or two more quick questions. What are the pH and conductivity tolerances for implementing consistent step gradient?
Yeah, for pH tolerance, I would say ideally, it will be the best to be in 0.05% tolerance range. So having, let's call it 7.5, that is 7.45 or 7.55. But we also then we know that this is quite a struggle to get to this values. So usually 0.1 tolerance is also acceptable. Definitely 0.2 will then you sacrifice either the purity or recovery. So 0.2 is a far shot, but 0.1 is manageable, and then anything below that, great. As far as for conductivity, we do see that that 3% this is also something that we used here in this case study that I shown, basically showing that 3% tolerance will put you in the linear range for especially let's say for challenging of the column with the different loads.
Perfect. We'll squeeze one more in. Can the loading amount vary during scale up?
So as Rok already shown, it's really important to keep the loading ratio per milliliter of column in a range that is acceptable, and so that is defined during the development. This range can be, we've seen like a 1 log difference, but we need to maintain in these numbers to keep it consistent.
Thank you, Rok and Maya, for answering those questions. That is all we've got time for today. Any questions that we didn't get to, we will reply later by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left then is to thank Rok and Maya once more for a great presentation, and thank you to the audience for listening. We do hope you'll join us again soon.